All right.
All right. Welcome everyone for the first seminar in 2026. And thank you very much, Michael, for agreeing to give the first seminar. And it's all yours.
Thank you. My pleasure. I'm Michael Kay. I'm a professor of biochemistry at the University of Utah School of Medicine. And thank you, Kate, for the invitation to kick off the 2026 Build a Cell seminar series. I'm going to tell you about my lab's adventures in mirror biology. In particular, we have a strong emphasis on developing mirror peptide or D-peptide therapeutics. And so the image here, which sadly is not of my dog because they won't tolerate these costumes, but is to symbolize standard peptide therapeutics, the dog without the costume, and then when you switch the chirality to D-peptides, how these acquire some superhuman powers that make them a lot more durable in the body. And so I'll be telling you about our work developing D-peptide therapeutics, which is really how we got into mirror image biology. And then in the second part, I'll tell you a more of a synthetic biology story of how we got into the chemical synthesis of D peptides and proteins and developing new tools to try to make this easier so that we can consider making larger and larger, more complex mirror image machines to ultimately build up more mirror image, utility in the toolkit. All right. And then finally, I just have a disclosure that I'm a scientific advisory board member for Eisen Therapeutics, which is developing D-peptide therapeutics. It's a startup. All right, let's dive in. So I just wanted to briefly start off with looking at the general drug discovery space very broadly. And so one of the big problems in the drug discovery field is that if you want to block a protein-protein interaction interface like shown here, this is a very broad, very large area interaction. It has traditionally been very difficult to do that. And so if we look at the general drug discovery space, there are the small molecule drugs that we all know and love. These are, things like aspirin. These are generally very cheap and easy to synthesize, relatively simple organic compounds. They are generally orally available, which is very convenient. But these molecules kind of lack the molecular sophistication generally to block these kinds of interfaces between protein-protein interactions. They really excel at blocking active sites or other small molecule binding sites on the protein that are naturally there, but to get into these big interfaces and interrupt them has been very challenging. And so people in the field often call these types of therapeutic problems quote, undruggable because they resist standard high throughput small molecule screening and medicinal chemistry campaigns. Coming on strong in the last several decades are biologics. These are biologically produced, tissue culture produced therapeutics, most commonly monoclonal antibodies, but can also be other recombinant proteins. These are much larger, and these do have the size and sophistication to really excel at disrupting these kinds of protein interaction interfaces. And so you've seen monoclonal antibodies really come on strong in recent decades for a wide variety of different applications in especially cancer, but many other areas as well. And so these are really ideal reagents, both in research and in therapeutics, for modulating protein interaction interfaces. But they are very large molecules. They're quite expensive to produce, and they often trigger an immune response themselves, such that, especially for chronic diseases, you can get anti-drug antibodies so that they don't work as well over time. They're also quite bulky. A standard IgG antibody is about 150 kilodaltons. And so in some contexts, this could be a problem where they have difficulty getting access to small spaces. Somewhere in between and coming on strong in very recent years are peptides. So these are, smaller than proteins, but larger than small molecules. An example peptide is shown here. They're often in the, 5 to 40 amino acid range. And these also are very good at blocking these kinds of protein interaction interfaces. They can be made by chemical synthesis at metric ton scale, very cost effectively, as has been evidenced by the GLP-1 peptides that are really revolutionizing medicine right now. And because these are synthetic and can be done at really large scale, they're dramatically cheaper to produce than monoclonal antibodies or other recombinant proteins. And because they're so much smaller, they are more nimble. They're able to diffuse into solid tumors or get into other narrow spaces that antibodies may have difficulty accessing. But the reason why peptides are not everywhere in the drug therapeutic space is primarily because of their fragility. They are extremely vulnerable to proteolysis in vivo, and in some cases only last for a couple of minutes. And if that doesn't get them, because of their small size, they are typically filtered by the kidney very rapidly. And so this can give a very, very short half-life that makes them extremely challenging to give therapeutically, especially for chronic diseases. So we're inspired by what peptides can do, but frustrated by their limitations. And so our lab has really made a dive into looking at mirror image peptides. And so let me introduce you to those. So just a very basic review about what dipeptides are. They're the same as natural peptides, short amino acid strings, except they're composed of D amino acids instead of the natural L, the mirror image amino acids. And so these do not exist in nature, so they only can be made chemically. And the key benefit that leads to other benefits is that they are not recognized by proteases. If you look at the active site of all of the major degradative proteases in humans, they require an extended conformation of the L chirality in the backbone in order to be recognized. And so if you put a dipeptide in with proteases, essentially nothing happens. They're not a good substrate. And so this has the potential to allow you to use much lower and much less frequent dosing at a much lower cost. It also turns out that dipeptides are very poorly recognized by the immune system. And a big reason for this is that part of the maturing immune response to a standard L peptide is that they're often chopped up into pieces for presentation by the MHC system, which allows for a more optimized immune reaction. And because they can't be proteolyzed, dipeptides resist that process. And so very often these trigger, no or minimal immune response, another nice benefit. So this sounds really great, but how are we going to actually access dipeptides? There aren't any natural examples to inspire us. So how are we going to develop and design them? And so the main technique that we use is a really beautiful technique that was developed in my postdoc lab, Peter Kim's lab, before I arrived, called mirror image phage display. And let me just briefly walk you through that here. So shown schematically on the right here is a bacteriophage, or phage for short. It has this tail protein shown in green that's on the outside of the phage. And what you can do is fuse a genetic library of randomized DNA sequences to the tip of this gene 3 protein that displays on the surface of the phage. And so if you clone a library, often of billions of different members of random peptide sequences fused to these tail proteins, you can then throw that group of phage onto some selection system, say binding to a target, and go through a few rounds of selection and amplification, and then ultimately sequence what are the ones that are enriched. And that will tell you the sequence of the peptide on the tip that bound to your target. So that's standard phage display. Got the Nobel Prize for George Smith about 10 years ago and has been involved in a lot of drug discovery and development. Very powerful technique. But that's for standard L peptides. Mirror image phage display is shown here on the right, where we do the same kind of thing, except now we chemically synthesize the target we're interested in out of D amino acids. If you do that, it will fold up to the identical 3D shape as the natural protein, but mirror image. Then we select for the L peptides on the natural phage that bind to our chemically synthesized mirror target. And by the law of mirror image symmetry, this is a mirror here in silver, if an L peptide on the phage binds to the mirror image target, it must also be true that the mirror image of that peptide, now a D peptide, would bind to the mirror image of this target, which gets you back to the natural L target. So we use this symmetry trick to get around the fact that we can't make D phage yet. And so this is one of the few things in biology you can count on. It's the law of mirror image symmetry, and it works perfectly. And you can use this to develop D-peptide therapeutics. There is one catch, though, in terms of the practical widespread applicability of this, and that is you have to be able to chemically synthesize your D target, since these cannot be made using standard recombinant methods. And that's often the rate limiting step in these projects. So I'm going to walk you through a long story that started back when I was a postdoc and has continued to the present day of how we apply this to HIV. And this is really our entry into the whole field of mirror image therapeutics. And so we're going to describe D-peptide inhibitors of the HIV entry reaction. And I just want to acknowledge my longtime collaborators here who have helped us over many decades actually now to develop and bring these peptides to the clinic. So first, I just want to give you a very brief review of how the HIV entry mechanism works. If you've seen other viral fusion or other membrane fusion pathways, they're all very, very similar. They're all variations on a theme. But HIV is one of the best studied ones and the one that we're targeting here. If we look at a virus shown on the bottom here, here's the viral membrane. It has this envelope spike protein on the surface that forms these trimeric nubs. And this protein is composed of two subunits, a transmembrane subunit called GP41, and then a surface subunit called GP120. And here's a cell membrane of a cell that HIV is about to attack. GP120 recognizes CD4 and one of a couple different chemokine co-receptors and undergoes an activation process, shown here as a subtle tilting of GP120 to the side, that activates GP41, which contains the actual fusion machinery. Once this activation happens, GP41 unfurls, as shown here, and then has this red region called the fusion peptide that embeds, harpoons the target cell membrane, and then at that point has attached the virus to the cell, but hasn't gotten in yet. At this state, which we call the pre-hairpin intermediate, there's an N-terminal region of GP41 that forms a trimeric coil like shown here that we call the N-trimer for short, and then a C-terminal region here that connects it to the rest of the virus. And in a slow reaction, this N-terminal coil and the C-terminal peptide, these have a very strong anti-parallel affinity for each other and collapse to form this 6-helix bundle complex. And the collapse of that, which we call hairpin formation or trimer of hairpins formation, pulls the two membranes into close apposition, and that's what drives membrane fusion. And so this is how HIV ultimately infects and enters a cell. So it's a process we would like to very much inhibit to prevent HIV infection. A key milestone in proving this mechanism was finding ways to inhibit it. And it turns out that if you take a peptide that corresponds to the C-terminal region here and just make it as an exogenous peptide that's called Fusion, shown here, a 36 amino acid peptide, this actually blocks fusion by binding prematurely to this N-trimer into those grooves and then competing with that hairpin closure. And so since the hairpin can't close, the virus can't enter. And so this was a very important new drug in the very, very late 1990s and early 2000s. At the time, I think was one of the fastest drugs ever to be approved because there were a lot of challenges with limited HIV therapeutic options at that time. If you look at this N-trimer a little bit more carefully structurally, we're zooming in here, there's a region right at the base there where the red arrow is, that's this very deep hydrophobic pocket that seems to be especially important for this hairpin closure. It's extremely well conserved among many HIV strains. When people saw this structure, there was a massive worldwide effort to develop a small molecule that would bind to that pocket and block fusion. And unfortunately, this ended up being one of those classical undruggable targets. A lot of great work went into trying to find good drugs against this target. And I'll just summarize that work by saying it was very, very challenging. It was hard to find molecules that behaved well, that weren't essentially denaturants or insoluble. And so it became a classic study for an undruggable drug target. So let's talk about Fusion in a little bit more details. Fusion has a low nanomolar IC50, which is quite good, this was the first FDA-approved C-peptide that knocked down viral load by a factor of 100 almost immediately. And a startup company, Trimeris, that was ultimately bought out by Roche, took over this and brought it to approval. But there are some really tough practical limitations to Fusion that meant that it was never a widely used drug. The big one is proteolysis that I mentioned before. It has a very short half-life in the blood. And so the way that they overcame that is by just giving massive doses of the peptide to overcome this very short half-life. That meant 90 milligrams twice a day by injection. And this ends up being about 70 grams of peptide a year, a truly massive dose, very, very high cost. It's actually very hard to inject that much peptide across the skin. There are a lot of injection site reactions. There were solubility challenges. This peptide came as a dry powder, and patients had to reconstitute that powder every day with sterile water. And it took something like 45 minutes of stirring and shaking to get it to go all into solution. And if you didn't get it to go all into solution, you would inject hydrophobic globs that could cause worse injection site reactions. So it was a very inefficient, very difficult drug to take, but it was life-saving at the time for patients who had no other options. Unfortunately, resistance developed very quickly. It turns out that this peptide was discovered by kind of a random screening effort of different fragments of HIV, and it misses that very conserved pocket region. And so, unfortunately, it was much more vulnerable to resistance than would be optimal. And so, as a result, Fusion was never widely used and has actually been discontinued, but was an incredibly valuable proof of concept that entry could be a valuable target if only we could find a more practical way to access it. And so that's where we come in. We wanted to develop a dipeptide pocket inhibitor of HIV. And so I'll summarize a ton of work over many years here because it's all been published, but just to kind of get you up to speed. So we did mirror image phage display, as I described earlier, using a design mimic of that N trimer region of GP41. And that gave us some mid-micromolar inhibitors against a lab-adapted HIV. These are easy to inhibit HIV strains that we start off with before going to clinical isolates that are more challenging. And those initial hits all had a very similar consensus sequence that's shown here that kind of got us going in the right direction. And this is a crystal structure of that first D-peptide inhibitor binding to that pocket region of GP41. And this was the first proof that if you target the pocket alone, this is enough to inhibit HIV entry. So it was all very promising, but not really in the potency range you would need for a real drug. And so we used this crystal structure and the sequence data we got from that initial phage display to do affinity maturation, doing constrained libraries that fixed the residues that were critical for this interface and allowed other residues on the backside to float in a library to optimize the sequence further. We were helped quite a bit by Chris Hill here at the University of Utah, who's a wonderful structural biologist who helped us to get structures of all these different D-peptides and to understand how we might go about trying to improve their affinity. And so I'll skip over a ton of work here by saying we went through a few cycles of additional phage display and structure-assisted design to ultimately end up with what we called PI12, or pocket-specific inhibitor of entry number 12. Only took 12 tries. Here's the sequence shown here. It has a disulfide bond shown between these two cysteines that gives it a little bit of rigidity and compactness for fitting into that pocket. And this is now more than 1,000-fold improved compared to our original inhibitor into the low nanomolar region, getting a lot more intriguing for potential therapeutic use. And then a really big breakthrough is once we had that nice monomeric dipeptide, we realized that our target is a trimer. And so that pocket is in the groove between neighboring alpha helices in that N trimer. And so our structure told us exactly how those dipeptides sit relative to each other. What if we engineer these PEG spacers to connect 3 inhibitors together to get a trimeric version that would have a real boost due to avidity of binding to all 3 sites at the same time? And this worked better than we could have even hoped and gave us Pi12 trimer, which had about 1,000-fold even greater affinity boost. And in fact, the affinity of this is now so high that we struggle to measure it accurately. It's just too tight. So at this point, we got really excited that this really could be a drug against HIV. We had one more trick up our sleeve in developing this clinical candidate. And so if we look at the geometry of HIV here, shown very schematically, and where the N trimer and the C peptide are relative to a target cell, and then we zoom in, as you see here, we can see that this pocket region is about 60 angstroms away from the target cell membrane. And around this time, Merck had developed an interesting approach to a Fusion-like C-peptide inhibitor, where they added a cholesterol molecule shown in red here to loosely anchor the C-peptide near the target cell membrane and increase its effective concentration at sites of viral fusion. And so we wanted to apply that to our system, but accommodating this longer 60 angstrom gap between the pocket and the membrane. And so to do that, we had a lot of expertise in PEG engineering. And so we theorized that if you had a PEG 12 that was stretched out taut, that would cover the distance. But a more realistic PEG 24 would have an average persistence length that would cover this distance without having to be stretched out completely. And so we predicted that a PEG 24 would be ideal for this application. And so here's the construct that we made. We made a modular scaffold where we could hook 3 dipeptides on the right side here using NHS ester chemistry, and then a thiocholesterol on a variable fourth arm with different PEG spacers. And here's the data. We switched at this point to a clinical isolate, GRFL, that's harder to inhibit, which had a 2.4 nanomolar IC50 without any cholesterol. And then as we added the cholesterol with different PEG spacers, you can see that at about a PEG 24, we got the optimal IC50. And going longer than that was pretty similar, but if it was a little bit shorter, it wasn't quite as optimal. And this gave more than a 100-fold boost in potency. So schematically, you can see what this looks like here with the cholesterol anchoring the inhibitor on the target cell membrane in close proximity to the pocket. And as an additional benefit, cholesterol also extends half-life. This is something that's well known in the peptide field, especially by albumin binding. It just helps to reduce the filtration rate. And so with a dipeptide that's not vulnerable to proteolysis, this gave us a nice long-lived inhibitor that we thought could really be a clinical candidate. And so here's what our final clinical candidate looks like. We optimized some of the chemistry for large-scale production, and we call it CPT31 for cholesterol, pi12 trimer, and then 31 is the total PEG length that we ended up using to separate the cholesterol from the pi12 trimer. And we also work quite closely with a local startup company, Navigen, that's a University of Utah spinout, to do all the preclinical and clinical trials on this molecule. And so characterizing this molecule, the really important thing in HIV is resistance and breadth. There are so many different isolates around the world that have diverged quite a bit and have very different envelope sequences, although that pocket region is very highly conserved. And so this is a very complex figure, but let me walk you through it. This is looking at 118 different isolates from around the world for HIV, representing the different types of HIV that are prevalent in different continents. And all of these lines, except for our dipeptide, are broadly neutralizing antibodies that are in different stages of development. And so what we're showing is the IC50 cutoff on the x-axis, and then at that particular value, where you are on the y-axis is what fraction of those 118 viruses have been inhibited. So if you get up to 1 here, that means every single virus has been inhibited by about a 0.5 nanomolar IC50 cutoff. Things that are shifted to the right are less potent, and things that plateau at these lower levels never inhibit some of the strains that are in the panel. So ideally, you want something as far to the left as possible and that hits as close to 100% as possible. And as you can see, CPT31 does really well in this assay, inhibited all the strains tested at a sub-nanomolar concentration, dramatically better than any of the monoclonal antibodies that were tested. And in fact, CPT31 is even better than it looks here because we're using molar units for the IC50 cutoff here, and CPT31 is about 1/15 the size of an IgG antibody like the one shown here. And so if we used mass units, which is what's typically done with antibodies, this would be shifted even 15-fold over to the left. So this looked really promising for preclinical data and motivated us to pursue clinical development of CPT31. Next up, we collaborated with Malcolm Martin at NIAID, who's a world leader in monkey models, specifically rhesus macaques of HIV, and has developed SHIVs. These are simian HIV hybrids that mimic human HIV behavior for testing inhibitors and vaccine strategies. And so in his monkey model, you can have a rectal challenge with the SHIV and get 100% of the animals infected. And so what's shown here is the viral load over time of the control animals in black. And so what happens is you get a very rapid viremia in the first 2 weeks, and then it settles in as the immune system starts to control the virus partially, and you get to a viral set point. Very similar to what happens in humans. And this is a very high dose of virus that's being given so that literally 100% of the animals get infected. And so we wanted to evaluate our CPT31 in vivo. And so we dosed 3 milligrams per kilogram of CPT31 several days and several days before and after a high dose rectal challenge to see if we could prevent acquisition of SHIV. And in fact, we did. Here are the viral loads. These are all below detection. And so all 4 animals tested were completely protected. So we were quite excited by that. And then we just did it again with now a lower dose. And again, 0.5 mg per kg was also fully protective. And then finally, we dropped again to 0.125. And now we found kind of the limit of potency at about 0.125 mg per kg, which is a really nice low dose. So I'll just add, for the sake of time, we did some other studies, including a treatment protocol and a protocol where the animals were suppressed using standard HIV antiretroviral drugs and then maintained on CPT-31 and withdrawing that cocktail of drugs. So that's called maintenance of viral suppression. And the inhibitor worked very well as expected in those contexts as well. And so at this point, we had a pretty complete preclinical package and we were ready to go on. And so currently CPT-31, Navigen obtained the IND from the FDA, including all the toxicology data and large-scale production. And a phase 1 trial was completed in 2021. And you can see the trial results at this website. But the drug was shown to be safe. And these were healthy volunteers and behaved as expected in vivo. And so this drug is essentially now ready for phase 2 trials. We've gotten a little bit stuck at this point. Phase 2 trials are very expensive. And in the HIV drug space, there has not been a lot of investor interest in developing this particular HIV drug, which would have applications primarily in the developing world. And given the crowded HIV marketplace and the developing world market, we have struggled to find investors willing to push for a phase 2. So we're a little bit stuck at this point, but still looking around, still seeing if we can find someone to help us develop this further. Here's my old grad student, Brett, but now he's gone corporate after working with the FDA and working through Navigen. So looking ahead, the HIV field has really moved on. You may have heard in the news recently, there's a wonderful new drug that was just approved called lenacapavir, based in part on discoveries here at the University of Utah from Wes Sundquist's lab. And so this is a capsid inhibitor. And what's exciting about that drug is that the drug can be dosed every 6 months. And so the field is really moving to these more long-acting drugs. And so to help join that and to find companions for things like lenacapavir, we're exploring conjugating some of those broadly neutralizing antibodies to our D-peptide to create a very long-lived kind of a single molecule drug cocktail. And we're working with Pamela Bjorkman's lab at Caltech on that right now. We've learned a ton through this HIV drug discovery process about D-peptides, about mirror-rich phage display. And so we're now using this information to target other emerging viruses, particularly henipaviruses, which are pretty scary, group of viruses that cause a very severe pneumonia that can be 50% fatal and that, is spreading around the world at a low level right now. But if it were to become better at spreading human to human, could be the next big pandemic. And we really like to be prepared for that. And then to really introduce to the second part of the talk, getting more into the synthetic chemistry, this experience really encouraged us to want to expand D-peptide drug discovery to broader targets, including larger protein targets that had been inaccessible until recently because of the chemical synthesis problem. So let's get into the chemical synthesis problem. So the way we make D proteins and D peptides is by initially solid phase peptide synthesis. This is our peptide synthesis robot in the lab. Her name is Cynthia. Get it? And this uses very robust chemistry that has been around for over 50 years called solid phase peptide synthesis, where you have a resin bead in these reaction vessels at the top, and then you have the amino acids that you mix in and add them one at a time to build the amino acid chain on that resin. And this is extremely robust chemistry. It's done at industrial scale, and it works quite well up to around typically 50 amino acids, at which point you start to have problems with incomplete coupling, crowding on the resin, secondary structure formation on the resin that blocks the incoming amino acid, things like that. But really robust for smaller peptides. If you then want to go beyond those small peptides, we use a technique that was developed by Steve Kent's lab called native chemical ligation that's shown in the upper right here. And here you can take one peptide and put a thioester at the C terminus of it, which is easy to do chemically, and then a cysteine at the N terminus of a second peptide. These two peptides will then spontaneously react, as shown here, and you'll ultimately undergo this acyl shift that creates a native peptide bond between peptide 1 and peptide 2. And so this gives you a traceless connection. And so this is how we stitch peptides together. Now, cysteine is a pretty rare amino acid in protein sequences, so a big breakthrough from the Dawson and Danishevsky labs more recently was showing that you can actually do these ligations at an alanine by temporarily putting a cysteine residue in the junction site and then doing a site-specific desulfurization reaction that trims the sulfur off of this residue. And if you take the thiol off of a cysteine, that gives you just the methyl, which is an alanine residue. And since alanine is dramatically more common than cysteine, this gives you many more options for ligation junctions. And so I'm skipping over a lot of work here, but our big entry into this field was about 10 years ago now, where we made this very large protein, at the time, the world's largest synthetic protein called DAPA. It's 312 amino acids. And we made this because this protein is the smallest known protein that requires a GRO-EL/GRO-ES chaperone to fold properly. And we were interested in understanding whether that chaperone activity was stereospecific. And so to do that, we need to chemically synthesize this protein in D and then see if the chaperone could fold it. It turns out the chaperone can fold the mirror image protein, and so it has this kind of latent ambidextrous activity, which will be very useful for using those chaperones for folding future D proteins. But making this protein was really quite onerous. And so the way we went around doing this is we broke it up into these segments that are shown on the left here that are generally smaller than 50 amino acids. And then we have what we call the bracket, an assembly process by which we stitch them together using these NCL reactions bit by bit to ultimately create the entire full-length protein. But this is brutally difficult. There's a lot of inefficiencies here that I'll dive into in a moment. But once we did this, this took two of my most talented graduate students 2 years as their main project to make 1 milligram of protein using this technique. So really backbreaking work. And so here's their celebration when they finally completed this protein, well earned. So what made this so difficult, and what can we do about it, going forward? So taking a look at this bracket, peptide insolubility was our number one problem. It turns out if you chop up a protein into different segments of around 50 amino acids, in essentially every case, several of those segments are going to have horrible solubility. It makes sense that most proteins have a buried hydrophobic core, and if you take that out of context, you create something that's really insoluble. And that means that they're difficult to synthesize, difficult to purify, difficult to get to the millimolar level concentrations that you need to drive the bimolecular ligation reaction. And so that's often a dead end, and you have to try break up the protein some other way. That happened at all these places here with DAPA. Another problem is there's many, many handling steps here. All the sequential steps, all the purifications, all the protecting group removals, all of those involve loss of yield, especially when solubility is suboptimal. And so very often you would lose all of your sample at one of these intermediate stages and have to start over. There's way too many purifications, especially as you get to larger and larger intermediates. The yields of those purifications really decrease, and so it becomes very difficult to have enough material at the end to work with. It turns out that these ligation sites are highly variable depending on the amino acid right before the ligation site. And so some of the sites with bulky residues can be very, very slow, and you're competing with hydrolysis of that thioester, which is a dead-end reaction. And so that was another yield sapping problem with this. And so putting all this together made for the extraordinarily difficult synthesis of this protein. And so we really need to address these problems to make larger targets in the future for mirror image phase display and to do this more routinely. And we're inspired by Ronald Reagan's quote here about trying difficult things. So inspired by the DAPA story, we developed a series of tools over the years. And so let me walk you through the first one, solubility problems, with a tool we called Helping Hands. And we borrowed the Hamburger Helper logo here to help us advertise this. And so the idea here is we made a tool that could be attached to lysine side chain residues. And the tool is shown here. We attach this to a lysine residue on a peptide while it's being synthesized. And this gives us an extra N-terminus that is a branch that we can add highly solubilizing residues, like in this case, 6 lysine positive charges, in a temporary manner to allow us to work with the peptide, purify it, and then when we're completely done with the assembly reaction through ligation, this is a labile group that can be cleaved quite easily using hydrazine or hydroxylamine that doesn't affect the rest of the peptide, and then shaves that helping hand off to give you back the natural lysine side chain. And so what that looks like in practice is here's a peptide from the Shiga toxin that without the helping hand is this bottom trace by HPLC, where you can barely see any peptide at all. It's such a broad peak, we can't work with it. But if we add the helping hand, it really sharpens up that peak, and now we can really see the main component and purify it and work with it. So a pretty dramatic change with just one helping hand being installed. So this works great and is actually commercially available now for other peptide chemists to use. But it turns out that lysine is a pretty common amino acid, but there were situations we ran into where there weren't lysines around in insoluble segments. And so we developed something similar called a glue hand for the glutamic acid. And this time we made a pre-built amino acid with glutamic acid and a helping hand, again, that creates an N-terminus that we can add polycharged residues. And again, this can be cleaved using a palladium catalyst in a way that doesn't affect the rest of the peptide so that you can cleave the helping hand off after you're done with all of your handling steps, including ligation. And so here's again an example of an HPLC trace of a really difficult peptide in red without a helping hand that gave us essentially no signal by HPLC. But once we installed the helping hand, we get nice sharp peaks that we can now purify the correct component quite easily. So this has really helped us to address the solubility challenge. And because of how common lysine and glutamic acid are, about 12.5% of all protein sequences in the UniProt database, we can almost always find a lysine or glutamic acid where we need it in an insoluble segment. Next up, we dove into an informatics strategy to really look at these ligation strategies a little bit more systematically. And so we developed a tool called Alligator, or Automated Ligator. And the idea is you take proteins that you're interested in and you feed them to Alligator, and they will be chopped up into all the possible segments that are reasonable for peptide synthesis and ligation. And then you can score those based on considerations like the quality of the thioester residue, the solubility of the fragments, the difficulty of the synthesis, and then it gives you a rank-ordered list of the predicted optimal ligation strategies. And so this tool has been really helpful because, especially for larger proteins, there could be millions or even billions of different possible ways of breaking up the protein. And so rather than doing this by very tedious trial and error, this has really helped us to identify the most promising strategies, right from the start. And this is an open source tool that you can find on our GitHub site. It turns out that finding the segments is not the only problem. There's also a real challenge of the order in which you ligate them together and putting those brackets together. And so we addressed that with a tool we published recently called Bracket Maker. And so what this does is takes the output from Alligator and tries all the different orders of ligation. And again, makes predictions about which of these will be the most efficient in terms of solubility, protecting groups, and just general synthetic efficiency, and gives you a full synthetic pathway, like shown here, a full bracket that is predicted to be the most optimal for any chemical synthesis project. Again, this is available on our GitHub site. So next up, the problem I was mentioning about hydrolysis and just generally slow ligations is what we tackled next. And so we decided to pursue what we call templating methods, where we try to bring the two segments of interest together to accelerate the ligation reaction and minimize this competing hydrolysis. And so here we took the same helping hands we had developed earlier, but now rather than adding a solubilizing tag, we added these click chemistry handles. So shown here is a strained cyclic cyclooctine, and we're using that with a traditional azide click chemistry handle in order to click together two peptides via helping hands that are near the termini of the peptides we want to ligate. And if we do that, we can create this linked product here that brings those termini into very close apposition and gives you a really strong proximity-induced rate enhancement. And so we're calling this CAN, or click-assisted NCL. And how does this work in practice? So here's an example. We made a model peptide, Steve and Kent, in honor of Steve Kent's contributions to the native chemical ligation field. And so if we do the click chemistry through our helping hands on this peptide, if we just look at the reference peptides, non-templated NCL, we purposely picked peptides that had a slow ligation. You can see here, you only get up to about 40% ligation after 48 hours. And so that's a very unsatisfactory result that would be difficult to work with. But if we add our CAN reaction to the helping hands with no PEG linker, just making it as short as possible, we get these very fast accelerated ligations. And you can see that they're not terribly dependent on the concentration of the peptide, because this now becomes a unimolecular reaction rather than the bimolecular reaction of the untemplated version. And in order to simulate attaching the helping hand at sites that were further away from the N and C terminus, we added a PEG-8 spacer. And you can see that that does slow the reaction, as shown here, a little bit, but by only a modest amount, and it still goes to completion in a very reasonable timescale. And so this suggests that this can be quite readily accommodated by most peptide sequences. And in summary, we got over 1,000-fold improvement in the kinetics of the ligation reaction, especially at low concentrations compared to the non-templated rate, which will really improve the yield and allow us to work with more insoluble segments that we don't have to drive to such high concentrations. And as a bonus, it turns out there are certain ligation junctions. For instance, you can use that desulfurization trick with a thiolated valine, which is called penicillamine, except that it's very bulky and very, very slow. And with traditional solution chemistry, it's essentially impossible to get that to work. But with this templating, you can, with this proximity effect, force that reaction to happen with reasonable kinetics. So it gives you some additional ligation options that aren't normally available in solution. Next, we started to get a little bit greedier. What if we want to extend this to 3 or more segments? And so we called this the CAN-CAN, and I'll spare you the picture of us dancing the CAN-CAN to premiere this method at a conference, but that was one of our favorite jokes. The idea here is we introduced a pair of orthogonal click chemistries shown schematically here, where we do essentially a CAN of both peptide 1 and 2, and then peptide 2 and peptide 3, and then allow the ligation to proceed. And so there's a lot of chemistry we developed to get this orthogonal click working, but I'll refer you to the paper if you're interested in those details. But when we run the reaction, we got a little bit of a surprise. The correct peptide product we wanted, which is the 3 peptides in a row, product A, is there in a good yield, much better than we would get in solution. But it turns out that there are pretty serious competing side products. One of them is peptide C here, where this central peptide reacted with itself to create this looped peptide. That's a dead-end product. And we also had crossover between peptide 1 and peptide 3, where we got this peptide 1,3 product, which is also a dead-end intermediate, and that's shown here. And these are in pretty substantial amounts, creating a challenging purification. So to overcome this issue, we developed a more complex method that we call CAPTAIN for controlled activation of peptides for templated NCL. And this was published recently in Angawanti Chemie. And the details of the chemistry are not so important here, but I'll just say that we have 2 different thioesters, one called NBZ and one that's a standard thioester here, hydrazide thioester. And the way we work this is it's very similar to what we did before, except now we selectively activate one of the thioesters and then the other sequentially, so that we can control the sequence at which these ligations happen. And so if we do that with the same reaction I was showing you earlier, now we have essentially completely eliminated that 1,3 product and greatly reduced the cyclization of the central product, creating a much easier to purify dominant desired product. So this is looking really exciting, and we think is going to be incredibly helpful. Looking ahead now, we're really interested in trying to expand this to an arbitrary number of peptides. And so this is work very much in progress that we call TEMPO for templated peptide oligo NCL. And the idea here is we're using our helping hands now to attach oligonucleotides such that we have a master oligonucleotide template and then use those oligonucleotides attached to the helping hand of each of these peptides to find their spot on that master template such that all the peptides are adjacent to the peptide that they're going to ligate to and allow them to ligate in one cooperative step in a big assembly line. And so the chemistry of this is quite complex and we're still working it out, but we think this would be a great ultimate solution for making arbitrarily long mirror image proteins or other synthetic proteins without having to have all the intermediate purification steps. And so this could really unlock much larger targets in the future. So looking ahead and how we've been using these tools, we really are quite diving into larger D protein applications right now. And so there's one category of targets that I like to call gutsy targets. Gutsy both because they're relatively large and because they're in the gut and involved in gut pathology. So we published one target, the Shiga toxin, that is involved in causing severe diarrheal disease associated with Shigella. And TNF alpha is a heavily implicated inflammatory factor involved in inflammatory bowel disease as well as rheumatoid arthritis and other inflammatory conditions. And so we've been able to synthesize both of those using these techniques I've described. We haven't published this yet, but we just finished a target from uropathogenic E. coli, or EPEC. This is the pilus shown here. It's this tip that is expressed by these bacteria that allows the bacteria to grab onto the urinary tract epithelium and hang on during urination that normally clears out bacteria. And so this allows these bacteria to cause severe urinary tract infections and crawl their way up the urinary tract. We're trying to inhibit that tip so that these bacteria would get flushed away in the presence of a D-peptide and would be rendered non-infectious. And we just finished that synthesis recently. And then very exciting, we just published this last month in Chemical Science, shown here. We made a mirror image version of streptavidin. And so streptavidin is a very well-known protein in biotechnology that binds with extraordinarily tight femtomolar affinity to biotin. And this is used for all sorts of targeting and immobilization techniques, widely used in diagnostics. But it turns out that if you make the mirror image version, it has essentially no interaction with natural biotin, but still has its incredibly tight interaction with mirror image biotin. So we're trying to use this to create an orthogonal binding system that can be used that isn't interfered with by natural biotin and with a mirror image protein that, for instance, could be given therapeutically without triggering a massive immune response. So we're exploring using this for drug pre-targeting, some proximity labeling applications, as well as diagnostic applications, where there's been a big problem recently with people supplementing biotin, and this interferes with the biotin-streptavidin interaction and the ELISA assays that are quite widely used in diagnostics. So if we had the mirror image version, this would be immune to that kind of interference. A long-term goal of ours for a long time has been to eventually get the chemical protein synthesis efficient enough that we can consider making something as big as the D ribosome. And we're definitely not there yet. This is a huge complex with many large and complex proteins. But if you were able to do that, this would create a machine that people could use in the lab for making these mirror image proteins much more easily. But the central irony of this is that the difficulty of making this ribosome will mean that the peptide chemistry have to advance to a certain point that maybe you wouldn't need the D ribosome anymore. So this is a really interesting topic of debate and discussion in the field. Which brings me to my final slide. I just wanted to briefly bring up this really interesting effort that I'm sure you guys have heard about in this group, sponsored by the Mirror Biology Dialogues Fund. So in late 2024, Kate was one of the leaders in this, and I was one of the co-authors. We published a really interesting perspective article on thinking about mirror life. And I'm drawn to this because one of the arguments for eventually making mirror life is that you could use it as a factor for making mirror image proteins and therapeutics. And so if you read over this paper, we come to the consensus that mirror life is something that really is potentially feasible in the coming decades, but is something that we should really seriously consider not doing because there are some very poorly understood, potentially very serious risks in terms of biosafety that really we can't control or can't really fully evaluate right now. And so as a result of this initial call and a series of conferences sponsored by Mirror Biology Dialogues Fund and others, there's been a lot of discussion about how to handle a situation like this that's still, a decade or more away, but that probably would happen by default if we don't do something active to regulate or limit activity. And that debate and discussion is really ongoing. And a really central part of that discussion has been whether the D ribosome itself is a key enabling technology that needs to be prevented or regulated, or whether that would be a stopping point that would still provide utility, but wouldn't have to progress to a self-replicating system. And so this debate is very much ongoing. If you're interested in this very interesting topic, I recommend you check out the Mirror Biology Dialogues site. There have been many different workshops and conferences, and we would love to hear more from the community about how people are thinking about this very complex and interesting problem. And so with that, I just want to give acknowledgement to my DREAM team, our funding sources, and thank you for your time. I would be delighted to take questions.
Thank you. Thank you so much for an amazing talk. There is a lot of questions in chat. Can you see chat now?
Let me stop sharing my screen and then I should be able to. Okay, yes. Great.
If you can read a question, because people watching the recording won't see the questions.
Great. Happy to. I'll take them in order. Okay, so when you make a peptide with mirror amino acids, how does it get metabolized? Can anything break it down? As far as we know, no. We've taken D-peptides and put them in blood samples for a month, and we come back and they look the same. There is some slow degradation due to environmental factors like UV light, oxidative damage, things like that. But as far as we could tell, there's no natural metabolism that can deal with them. In our case, we purposely kept our D-peptide relatively small so that it's ultimately filtered by the kidney into the urine unmetabolized because we don't want it to accumulate indefinitely in the body. So that's how it leaves the body. But as far as we know, there's no metabolism going on. What aspects of the Pi12 discovery process were uniquely enabled by protein engineering approaches, and which steps would have been impossible using traditional small molecule drug discovery? So almost all of it was related to evolutionary, phase display, directed selection, and then later structure informed, optimization, which on a peptide is kind of like medicinal chemistry, but it's much more structure informed than medicinal chemistry is. And the number of compounds that you're making is a lot smaller. You don't typically make thousands of peptides in the way that you do with small molecules. And it's a little bit more predictable, especially with current AI-informed design tools. So it was actually, pretty unique with the directed evolution, approach. But as you start to mature, it gets more and more like a small molecule medicinal chemistry process. How does targeting GP41 with Pi12 overcome the resistance pathways that limit many existing HIV entry inhibitors? Very good question. It turns out that pocket is perhaps the most conserved region in all of HIV. And so it's extremely difficult for that pocket to evolve away from our inhibitor. And there's a couple reasons for that. One is it has this kind of like a clasp on the hairpin that's really important for viral function, and so it's reluctant to disrupt that clasp. But additionally, the RNA that encodes that region is a structured RNA that's part of something called the Rev response element that's involved in the export of the genome and its packaging. And so even silent mutations in that region can disrupt that RNA structure. And so it just has a lot of constraints on it. Eventually, it does develop resistance, but at a very high fitness cost to the virus. And like all HIV drugs, this is something that you would use as a combination with other drugs, with other mechanisms to further add to that genetic barrier. To what extent did D-amino acid incorporation improve Pi12 stability and half-life compared to an all-L counterpart? Pretty dramatic. So Fusion, I don't recall its exact half-life, but it was on the order of a couple of hours, maybe up to 4 hours. And our D-peptide is about 24 hours. And it's all renal filtration, not degradation. So pretty dramatic benefit. If you look biochemically, again, letting it sit in a blood or serum sample, the D peptide lasts essentially indefinitely, and the L peptide, falls apart in a matter of minutes to hours. What adaptations, if any, were required to apply native chemical ligation to fully D-amino acid peptides? Were the differences in ligation efficiency or side reactions compared to L-amino acid systems? Good question. No difference at all. It turns out that there's no real, chirality difference in basic chemistry. If both components are the same in terms of inverted chirality, everything is exactly the same. The one practical difference is the reagents that are available in the D-amino acid space are much less common than they are in the L-amino acid space. So if we need like a modified amino acid or something special, we have to make it ourselves rather than just getting it commercially. And so that adds a little bit of a challenge. I will also say that the purity of D-amino acids is not as good as L-amino acids. They're quite pure, but the L-amino acids are amazingly pure. They're kind of biologically purified versus the D that are chemically purified. And so there are subtle contaminants that accumulate in mirror proteins of the wrong enantiomer. Even, 0.1% of the L-amino acid over 1,000 amino acid, protein is going to accumulate where, every protein has a defect. So that's a challenge that is unique to D. Okay. Were any backbone-dependent conformational constraints in the D-peptide fragments found to limit effective intramolecular S to N acyl transfer during NCL? So, not really. This is really pretty universal, but there are some steric issues that do pop up. There are some residues either at the N terminus or the C terminus that sterically we don't think it's actually the S to N acyl shift that's being affected. It's primarily the initial, thioester reaction. And so those are reactions that often are quite slow if they aren't templated. How long does it take you to synthesize large dipeptides now? So there's a hierarchy here. I'd say anything that's 100 amino acids or less, we can do in a couple weeks. Say under 200 amino acids, of an averagely behaved protein, maybe a month. And then above that, it still is a bit of a research project. And so I would say it could take, 3 months to 3 years for things that are larger. And anything above about 300 amino acids is still pretty challenging. The way the field has often dealt with this is if you have a protein that can be broken up into complementing fragments that then spontaneously assemble together, this has been a great way to make some of the largest proteins available. And so this is how like a 700 amino acid protein was made, not as one segment, but as several that complement together. But that's very often not possible and can be challenging. Polite thank you comment. Thank you. How expensive is it for you now? That is a great question. D-amino acids are actually not that expensive. It's really time and labor that's the expense here, and the solvent and the columns and all of that. We estimated that DAP-A cost us about $100,000 to make 1 milligram, and so a little pricey. I would say that's probably down by a factor of 10 to 100 now, based on what we've done so far, but still quite expensive. And so this is where the D-ribosome comes into play. If we're ever going to have therapeutic proteins like D-streptavidin that we make at, gram scale at therapeutic quality, that's something that might be really challenging to do by synthesis methods alone, and having a recombinant method, could be quite helpful. But I think that's very much an open question. Cool. And then from Pete Carr. Hey, Pete. Would I be willing to conjecture on what future experiments might help us better assess the risks of mirror microorganisms? Yes, I think this is a topic that's come up at a lot of these conferences and discussions. That's an important one. To me, the thing that we understand the least is how a mirror organism would be affected by a huge surplus of mirror relative to its metabolism resources. So for instance, it's pretty easy to show, that an organism can grow on achiral substrates pretty effectively, but it's not really been carefully studied to see if having like a huge excess of D-glucose for a mirror organism would actually interact in subtle ways with the metabolism to actually be a poison and compete with the L-glucose that would prefer to use. And so I think that's a really important unanswered question for thinking about, how viable could something be? I think this really great data showing that a mirror organism would certainly not just die. That it could grow on ACOL reagents that are widely available in nature. So I think that's an important one. And the other one, this is actually a study I've been trying to get funded for many years, but without too much luck, is trying to do a directed evolution experiment where you purposely try to take E. coli and grow it in a simulated mirror nutrient, universe, and see how quickly does it adapt and what are those adaptations? And is there much of a barrier, or is it really just quite easy for it to incrementally get better and better until the point where it has all the racemases it needs and it can grow fully? And I think seeing how hard that would be would really help us to evaluate the risk, or maybe if someone in the future wanted to do this, which I don't think is a good idea, create, more crippling, design flaws in a way that would cut off those evolutionary pathways. Although I think that's going to be, it's a little bit arrogant to think you can cut off all evolutionary pathways to create a truly safe organism. I think that's the end of the list.
I just want to say that the experiment you mentioned, that's something I've been dreaming about too, and predictably no one seems to be interested in it now. There is one more question that Elibio posted, and I think he posted in a private, not all channel, so I'll just read it. Do you foresee possibility to use large D protein applications fused with D or L peptides to reduce toxicity and control RNA viruses?
So I do think that there's a lot of targeting work, especially in the antibody drug conjugate field, that's quite exciting and really starting to take off. And I think there's a lot of room for using dipeptides as a reagent in that kind of context. What's really difficult about especially antibody engineering is that the more engineering you do on an antibody to make bispecifics or conjugates, you create things that are looking less like a natural antibody and tend to attract more of an immune response. And so some people call these Frankenbodies. Using a mirror image reagent as a oligonucleotide or a dipeptide as a targeting reagent is something that in theory could minimize that additional immunogenicity, and I do think has real potential. And so it is something we're pursuing with HIV as an antibody drug conjugate, but I do think that there are a lot of other potential applications there. Nucleic acids are even more vulnerable to rapid degradation by nucleases than peptides are. And so there are, companies that are working on, mirror image aptamers, things like that, that I think have a lot of potential as well.
Thank you. Thank you so much. And thanks everyone for great questions and a great discussion. And thank you, Michael, again for a fantastic talk. That's a great way to start a year.
Thank you. Thanks again for the invitation, and real pleasure talking with all of you.
Have a great rest of your day. Bye.
Bye-bye.